everybody. Welcome. It's absolutely great to have you here today. We're going to be looking at the science of ovarian aging and why it matters to longevity. Before we start today, we're going to give a brief introduction to ourselves and how we became interested in ovarian aging. So as your host today, I'll start. My name is Dara Campbell, and I'm head of research at Longevity.Technology's Market Intelligence Unit. Our goal at Longevity.Technology is to continue to be the leading destination for longevity innovation, information, and investment reporting. We created our new Longevity Market Intelligence Unit to focus on delivering high-quality market reports to really drive the longevity economy. And the latest report that we published was on ovarian longevity. I learned so much about the impacts of ovarian aging and the scientific research that's happening today, which could be translated into products in the future, which I'm hoping to dive into more today. And I think my interest in ovarian longevity really came from when we were contacted by some researchers about their work that they did with melatonin in ovarian aging. And I was just shocked as a geneticist and having done 2 degrees in biology that I knew nothing about women's health. So it's absolutely fantastic to have Dr. Jennifer Garrison and Dr. Keshav Singh with me today. They're both doing incredible work in the ovarian longevity field, but my introduction does not do them justice, so I'm going to let them introduce themselves. Dr. Jennifer, could you please tell us a little bit more about your background and how you started in ovarian aging?
Sure. Thank you so much for having me. I really appreciate the opportunity to talk about this stuff. I love having these conversations. I am a professor at the Buck Institute for Research on Aging. I also have appointments at UCSF and at USC, but you know, my training was actually in chemistry and chemical biology. So I got my PhD at UCSF working on finding the target of natural products and doing a lot of sort of drug discovery type work. Then I switched into neuroscience for my postdoc. I was really fascinated by bioactive peptides, and I moved into studying neural circuits that control behavior. When I opened my own lab at the Buck Institute, I was really passionate about studying aging and trying to understand what are the basic molecular mechanisms that govern aging in a human. And as a neuroscientist and someone who's really excited about bioactive peptides, I focused on trying to understand how changes in homeostatic circuits in the brain, so basically the parts of the brain that control things like energy homeostasis, fluid homeostasis, body temperature regulation, circadian rhythms, but also reproductive function, change with age. And this part of the brain is kind of special in that it uses chemicals to signal to distant parts of the body, right, in both directions. And so that's how I came to reproductive aging. We were studying one of these bioactive peptides and really digging into the mechanism by which it was working. And we came upon reproductive aging. And so, you know, thinking about central nervous system control of reproduction has gone on for, you know, at least half a century. But it's been kind of siloed and really, I would say, narrowly focused, and we're trying to change that narrative. So we're studying the neuronal pathways that control female reproduction and trying to identify the factors that lead to the onset of reproductive decline in women.
That's brilliant.
So
Thank you. Yeah, so exciting. I love your work. And Dr. Keshav, moving on to you, can you tell us a little bit about your background and how your work is related to ovarian aging?
I think I have to ask you how far I should go back. Okay, because no, I'm just kidding. So I can tell you, so my undergraduate was in India. I work, believe it or not, with cow dung to make methane, and my brother made biogas and lit the city in India. So I came to Australia. I was more interested in marine biology at the time. I got more interested in extreme life forms, came to Woods Hole for 3 months in a summer program, and he stayed up there for 9 months. And then I fell in love with the Boston area and all that. So I went back, finished my PhD, came back as a postdoc, and my postdoc was in DNA damage and repair. Came to Hopkins as a faculty, and I was looking for a niche where I can make some significant contribution and find my own way research areas. And turned out when I was looking for DNA damage repair, there was nothing known in the mitochondria. But that was a mistake at the time because there was a recognition that there is no repair in mitochondria. There were very senior people who have published paper, but turned out there is no repair for UV-induced lesions, which is very relevant for skin and other things. But anyway, that led me to get a little more determination in a sense, because people were saying, well, I think you were done. So I said, no, I'm not done yet. So I started a journal called Mitochondrion. I started a Society for Mitochondrial Research in Medicine. I edited a book before I could get my first grant. So it was quite an educational thing. And then I found the gold mine, because there's a set of mitochondrial diseases which are unrecognized, still happen to be. And then I worked with the Mitochondrial Disease Foundation, and that led to a lot of other things, congressional caucus we have, and I became an activist in mitochondrial disease. So we work around the world. And as we were working towards it, and what we found at the time, and so there are children and women in particular who have mutations in polymerase gamma, and that affects mitochondrial DNA, mitochondrial function, and they show clear premature signs of ovarian aging. So in last couple of years, when we developed this platform where we can disrupt mitochondrial function in any desired tissue, and the work we have done is related to whole body dysfunction in mitochondria, we had started looking at ovarian aging, and particularly premature ovarian aging, and we could talk more about it. And it just happened that I was looking for someone, and a clinical fellow, Jasmine Chiang, she walked in my office and said, hey, I want to work on your mice. And I said, well, you are a clinician and never worked on the mice, so how could I trust you that you, but well, she was wonderful. She's have a clinical practice. She's a fertility clinician. And we got really interested into it in the last couple of years, and here we are.
Brilliant. Thank you so much. Well, it's absolutely brilliant to have you both here today to talk about ovarian longevity. And I think we should just get straight into it. So first of all, we kind of all know women hit menopause at different ages. And, as a female myself, you kind of have the standard as we age. At some point, we're going to hit menopause and our fertility goes down. But I don't think there's kind of like a lot of knowledge around how that actually happens. And one of the things that kind of shocked me when I was doing my research was the impact of quality and quantity on a female's fertility and how that contributes to menopause. I was wondering if each of you could, describe a little bit more about how quality and quantity changes with age and kind of the pathways that are involved in this process. And Dr. Ketchab, we'll start with you if that's okay.
Yeah, so I look at, from the mitochondrial point of view. So mitochondria are one of the 9 hallmarks of aging in general. And what we have done is connected the mitochondria to other 8 hallmarks. So, for example, mitochondrial dysfunction, we have shown leads to genomic instability, leads to changes in intracellular communication. We were the first group to show that the mitochondrial effects leads to epigenetic changes, and including telomere attrition. So we have shown clearly that mitochondrial loss of function can lead to telomere attrition. Most of those applies to the ovarian aging and premature aging and menopause. Because if you look at so obviously when and that's something we have written quite extensively about it, as women age, their mitochondria becomes more dysfunctional. And one of the reasons is that they have low mitochondrial DNA content as they progress. They also seem to accumulate mutations, which we all do in other tissues, but it happens in ovaries as well. And as a result of that, ovarian, obviously the mitochondrial function, the things I just described, contributes to those. And one thing which people don't realize is about 2001, we wrote an article in Nature when that time they described the euplasmic transfer of mitochondria. We're describing what UPAS phenomena which women don't conceive, and fertility clinicians in New Jersey and other parts, they transfer the UPLAS. What we interviewed them, we wrote an article in Nature describing that euplasmic transfer contained close to about 500,000 mitochondria.
Mm.
So the reason we believe, and that's where I got more and more interested in developing a mouse model, because at the time when this paper was published, this turned out to be a three-baby story, three parents story. So that's how we got into it. And there was at the time, and even now, there's little or nothing in terms of having a model system to study the biology of aging in ovaries.
Yeah, brilliant. I mean, yeah, mitochondria is definitely something that came up a lot in my research into kind of the impact of aging on quality. And it's amazing because it happens first in the ovary. It's kind of the first signs of where that mitochondria is starting to decline. And it's because, oocytes depend so much on mitochondria during their development. Dr. Garrison, have you got anything to add kind of on the quality and quantity with age?
I do have some things to say about this.
Brilliant.
Yeah, so I mean, this is what's known, right? Is that menopause is essentially, the hallmark of menopause is that it's when a woman runs out of eggs. And we're all familiar with this graph of egg number and egg quality over time, which is essentially just characterized by a single inflection point and then a cliff.
Yeah.
Right? It's quite straightforward. But I think what we're trying to do is reframe the narrative around that graph. Is it really an inevitable truth? And I think the answer is that it's not that simple. So certainly egg quality and quantity declines with age. And the answer is that we don't know why. We don't know why that happens. We don't know what the cue or the timer is that sets that in motion, right? Ovaries are aging precociously. They're aging much faster than the rest of the body. And we don't know why that happens. So this is, the downstream consequences of infertility and menopause. We understand those, but why humans undergo this precipitous decline of fertility in midlife and what sets it in motion are a mystery. So when I introduced myself, I didn't mention that I'm the director for the Global Consortium for Reproductive Longevity and Equality at the Buck Institute, which is essentially trying to facilitate and accelerate basic science research in this area, because we want to know the answers to those questions. Our goal truly is to find the causal factors for ovarian aging so that we can intervene, right?
Yeah.
And to do that, we need to understand where along that trajectory that cue or timer happens. And we don't know whether it happens during development or, during puberty, early adult life, midlife. We're just not sure. And until we have those answers, I think everything that we're doing is kind of a Band-Aid. So in terms of number and quality, I think the age at which a woman goes through menopause is defined by that. But fertility isn't really, we don't think about it as a bank that only goes in one direction. It's stochastic. It's at the level of the individual, it's very different from person to person. And even within an individual, it fluctuates and changes. And so it's not necessarily this single inflection point in a cliff. It's much more complex than that. And I think we need to treat it as a more multivariable sort of phenomenon when we study it.
Yeah, I totally agree. And I think it was mad when I was doing my research at just kind of the varying numbers that they give when they say kind of women are born with this many eggs, and it varies by 2 or 3 million. And that's a big difference. And you're like, oof, okay. And then that number again decreases at the stage of first menarche. And again, the number of eggs is totally different from woman to woman. And you can see kind of why there would be such a difference between women because of that as one factor. As you say, it's going to be a totally multifactorial thing that we are trying to get our heads around at the moment. So many people believe, and you've kind of touched on it there, Jennifer, about the ovary kind of being the pacemaker for female aging. It's kind of the first organ in the female body to age, and it ages very fast. But what does that mean for the rest of the female body when that organ starts to age? How does perimenopause and menopause then impact the health of the female? Dr. Garrison, we'll start with you for this one, if that's okay.
Yeah, absolutely. I think the easiest way to say it is that menopause makes a woman's body age faster. So it speeds up cellular aging. It changes a woman's overall health. So even in very healthy women, when they go through menopause, their risk of things like heart disease, cognitive decline, stroke, osteoporosis, depression, they all go up dramatically, exponentially. And so, beyond reproduction and fertility, the end of fertility essentially sets off this cascade of negative health effects. And, that's profound. It has profound economic impacts. It has profound, personal impacts on quality of life. And, long before menopause, even in young women, there is a link between health and reproductive organs and overall health. So this is something where, reproductive function and functioning of those organs can profoundly affect health in other parts of the body. So, it's something that I think maybe young women don't fully appreciate until they're right up on it. And older women, unfortunately, who have gone through it are all too familiar with the things that I'm talking about. Yeah. And, the equality portion of the consortium that we started at the Buck Institute, the equality portion is really focused on two things. One is fertility, giving a woman, democratizing reproduction essentially, and giving her control over when and whether she has a biological child. But also the equality portion has to do with that change in health after menopause, because, soon women are going to be living more of their lives after menopause than before. And so if we don't address reproductive aging, then essentially we're making gender inequality worse. So it has really major societal impacts as well.
Yeah, brilliant. I totally agree with all of that. Dr. Keshav, have you got anything to add?
Yeah, so what I like to add, one is that obviously the natural aging and leading to menopause, and as Jennifer mentioned about the associated effects of it. But I think we should also not forget that there is a 1 to 2% of the premature ovarian insufficiency in the woman. So if you look at just 1 to 2%, it's a very large number of women who are under 40 have early menopause and premature ovarian. So they are another population which we should think about how one can help and what can one do about it. What turns out to be interesting, something, again, I go back to how we got into it. So when Jasmine was working on it, and I told her, well, we need to go and find out what we know today in the field. And it turned out there are out of, in the premature ovarian insufficiency, there are close to that, exactly 8 different genes are identified. And those 8 genes happens to be all involved in mitochondrial function. So it's involved in transcription, translation, and the proteolysis, which is involved in that. So that got me really involved and so on. We have the tool, we have the motivation, and something we just have to keep working on it. So the platform, which we can talk a little bit more, how we have developed that and how we can address some of it, and at least the system we have.
Yeah. And I mean, I think the amount of women that don't quite understand the impact of menopause on their own bodies, which is not the fault of their own, but just we don't speak about it as a society. And actually kind of the work that both of you are doing, I think if women knew more about the impacts of menopause and perimenopause and how that impacts their lives, their work, their personal lives, it's something I can't believe not more people are talking about, to be honest.
I totally agree. I just wanted to say that's such a good point that you just brought up, that mitochondrial dysfunction is clearly so important for ovarian aging. But that brings up a really important point that I think a lot of people don't realize, and that is that all of the molecular mechanisms that we know about that impact aging in the rest of the body are also operating in the ovaries, but somehow they're just being triggered much earlier, right? So mitochondrial dysfunction, senescence, inflammation, DNA damage, all of these things that we think about as being important for aging overall are also operating in the ovaries. So it's, this is this is an important question to answer scientifically, not just for women, but for everyone.
Yeah, perfect model, a human model of aging. And I guess, to be honest, that's just gone straight into my next question. One of the things that I found with my research was a lot of the possibilities for ovarian aging are possibilities we're looking at in longevity research anyway, because the pathways are just so similar. It's the same things that are being triggered in the ovaries that are being triggered for overall aging. Could you guys talk about kind of the methods that we could use to regulate ovarian aging and even end menopause, which I mean, a lot of the longevity field will already know about. Dr. Keshav, obviously mitochondria is the one for you. Could you just talk a little bit about how we could target mitochondria and regulate ovarian aging?
Yeah, absolutely. So as I mentioned, that in the premature ovarian insufficiency, we recognize that, and we were working on the model to begin with for different regions as well, because I was interested in mitochondrial function in cancer. And believe it or not, the mitochondrial dysfunction is also the hallmark of the cancer. So we were trying to connect that how aging-related cancer, because 70% of the people who are above 60 get cancer as well. So we developed this platform where we can knock down mitochondrial function at will, at any time, at any age, in the mice, and that's what we are doing at the moment. And then the next step is, which we have already taken, is then our platform allows to make not only organ-specific knock down the mitochondrial function, we can do tissue-specific and cell type-specific. We put a fluorescent protein so we can go and isolate those cells. So we have done in what we call bicistronic promoter. So here you have a dominant negative gene, and then other side you have the GFP. So wherever this protein is expressed, the GFP will be expressed. We can go and isolate and ask the question. And in our analysis, we were amazed that the study we have done, and which is part of the UA portfolio as well, that we can recapitulate menopause, and we can look at many other phenotypes. I mean, for example, I can list some of that. So there is a uterine atrophy, there's vaginal atrophy. You see the markers of the menopause there. You see the small ovarian reserve. So all of those things are showing up that, well, this is just generalized knockdown of mitochondrial function. Now we can go in cell type a specific way. And as I mentioned, what fascinates me that why ovaries will have such a large number. I mean, on one estimate is like 5 million mitochondria. And atresia, is it part of that apoptotic pathway, which is something, or these mitochondria are so sensitive that something triggers it, and we don't know. So all of that questions are, in my mind, and I think we have the tools to address it. And obviously, we can do, therapeutic approach, because we can age these mice, and instead of waiting over the naturally aged mice for 24 months, we can do the rapid analysis of some of the drug screening and things.
Yeah, brilliant. Thank you. Dr. Jennifer Garrison, anything else you'd like to add?
Yeah, I guess I would say that I think we still have a long way to go in terms of doing the basic scientific research. That's part of what we're trying to do, is fund science in this area. But there are some really promising targets, I think, on the horizon, things like oocyte mitochondria for sure, and ovarian inflammation. Thinking about how ovarian aging can be accelerated by environmental factors, things like genetics, obesity, and other underlying conditions, like PCOS or polycystic ovarian syndrome, for example. So there's a lot of exciting research going on. I think the lowest hanging fruit and the things that will be realized first before we come up with a cure for menopause are going to be improvements in the diagnostic and the biomarker space. I think there's really a huge amount of room for innovation there. The diagnostics we have to tell a woman where she is in her reproductive span are rudimentary and crude, and I think there's a huge amount of space there. And then biomarkers, looking for biomarkers that will tell us not just where a woman is in her reproductive span, but to be able to watch things change, with trying to do interventions. I think that those two things are really exciting, and hopefully we'll be realizing gains in this space really soon.
Yeah, fingers crossed. So, I mean, I was going to ask you kind of what do you think is the most promising area for ovarian longevity at the moment, but I think we know the answer for you, Dr. Keshav, and obviously Jennifer, you've just mentioned a few there as well. So I guess I would like to ask both of you what you're working on kind of now and what's next for both of you in the ovarian longevity space. Jennifer, we'll start with you, if that's okay.
Yeah, absolutely. So my lab is really trying to define these ovarian signals that that the ovaries use to communicate with the brain and ask how those change with age. Honestly, I'm very biased, but I think that menopause originates from this disrupted signaling between the brain and the ovaries. Mm-hmm. So that's what we're working on. And separately from what's happening in my own lab, the consortium and the center at the Buck Institute, we're funding grants to spur creative research in this area. There's been a lack of funding and also a lack of data just around women's health in general. But we came into this space with the idea that we would try to build up the ecosystem to support research, to basically drive innovation as fast as we can. And so a big chunk of that is just trying to attract the brightest scientists to work on this problem. So we're funding grants. We funded 22 researchers across the world last August, and we'll have another call for grants soon. And those projects, that we funded a whole swath of space in this biology, trying to understand not just markers and diagnostics, but also looking for those causal cues, thinking about new model systems, because, we don't have really great models, as was already mentioned, for menopause, because humans and whales are the only species that go through menopause. So finding better models to use for testing interventions is a really big thing for drug development in this space. Yeah. And I would say that my biggest job right now is actually what we're doing here, which is just getting the word out, getting people to talk about what's happening, to think about this area as a place for innovation, getting people who don't normally talk to each other to communicate. So I spend a lot of time talking to people in private industry and clinicians and scientists, trying to sort of build those relationships in a way that will push things forward faster. So that's what's happening right now.
Amazing. Yeah, the work that you're doing is fabulous, and it's really pushing this field forward. So obviously I'm very grateful. And as you say, it's not just women, it's for everybody. So that's brilliant. Thank you. And Dr. Singh, what about you?
Yeah, so we take in a multi-fang approach to what I just described. So one obviously is the therapeutic approach on the diagnostic approach, and because diagnostic approach, one is still in infancy. People are using it for what they call microscore. So the microscore is used for embryo which don't implant easily, and if you have a high microscore, there's some problem, they don't seem to do well. So there are some aspect of that, but actually the one thing I do want to mention, as Jennifer said, so one aspect which we are forgetting is that up to 90% of the women who get ovarian cancer are postmenopausal. So I am committed to developing a model system with platform we have, and what is available only is the what's called a VCD, which is one of the epoxide, vinyl some epoxide, which gives you a phenotype which is close to the ovarian aging in women. So there's nothing out there in terms of understanding ovarian cancer, let alone the menopause and other things. So what our approach is at UA and in my academic lab. So academic lab is looking at more mechanistically how this signaling works. We're doing, for example, we knock down mitochondrial function in ovaries and certain cell types, and do RNA-seq analysis to see how the signaling is affected. But from the translational point of view at UA, we are doing FDA-approved drugs at the screening because we have the tool. and look at some other known and unknown. So we are using AI to give us, which can enhance mitochondrial function. So which we are using it for other conditions, for skin aging as one of the primary goals. But ovarian aging will benefit from what we do in those analyses as well. But so our approach is multifying. And in the lab, I can do my basic science, but I am more keen to invest more energy and time, and if any investment we get, in order for us to really propel this therapeutic approach. And we have ideas, and I think if we get the funding, we go for it.
Yeah, I mean, I know about Eva Biosciences and the stuff you guys are doing, and if anybody wants to check that out, their research is incredible. So this is my time to kind of ask if anybody that's watching has any questions, if they could just put them in the chat. And whilst they're doing that, I'm just going to throw a question, and if either of you would like to answer, then that's absolutely fine. So one of the things that kind of shocked me a little bit when I first started my research was there was a couple of people that told me ovarian aging has already been cured, was cured 5 years ago by ovarian tissue cryopreservation. And when I looked into it, it's the method where one of the ovaries is removed, sliced, and then placed back into the body, and that can be kind of extopically on the arm or in the uterus itself to preserve fertility and delay menopause. But I would just like your thoughts on that as a plausible method for women to delay menopause. And
Absolutely.
yeah.
I think about this a lot, and it's a really, so that it's not really even an experiment. That observation is what fuels my research. So the observation is that in women who have cancer, typically they'll freeze eggs before undergoing chemotherapy, because chemotherapeutic agents obviously damage the ovary. And but women who are too young, who are prepubescent or very close to puberty, they can't do that, right? So they can't retrieve eggs the way that they would from a woman who's older. And so what they do instead is they just take a piece of the ovarian tissue and preserve that. And what's been found, which is kind of amazing when you stop to think about it, is if you take a little slice of that tissue and then put it back, either, like you said, it doesn't even have to be in the ovary, it can be just under the skin or in a fat pad, put it back into the woman when she's older and wants to actually have children, that it rejuvenates the ovary in a way that essentially it's almost like resetting it, reprogramming it. Now, it's not a perfect technique because, it's invasive, first of all, to take a piece of the ovary. It's also not forever. So they have to continue to re-implant new slices to keep it working. But what this research tells us is that there is some cue that's coming from the young ovaries, and it's probably more complicated than just a single signal going to the old ovaries. There's probably signaling in both directions. There's probably some signaling to the brain and back. But this is why we're doing what we're doing, because I think it's not what, putting back pieces of ovarian tissue is not a general solution that's going to be applicable to all women, right? You would have to do it when you're 20.
Yeah.
So for those of us who are past 20, it's not a solution, but also getting a 20-year-old to do an invasive procedure and then pay to preserve that tissue and then pay to have that thing done. I don't think that's feasible, but it gives us this hope that we can find these factors and cues that will hopefully be able to reset things later. So it's really amazing stuff from my perspective.
If I may just add to it, so because I have another head which I work at the cancer area. So what happens is that and as Jennifer alluded, so the chemotherapy induced obviously the ovarian problems. Most of those chemotherapy drugs are lipophilic. So if you take cisplatin, for example, which is used for ovarian treatment, ovarian cancer treatment, or take doxorubicin or adriamycin, almost all of them are lipophilic, and they end up in the mitochondria. So our approach is to protect mitochondria in the ovaries and see if we can benefit from it. And it turned out that there are certain findings which will report it that one of the gene called MDR1, which is a transporter of this, and if you block that, I think your ovaries are protected. So our approach, as I mentioned, is if we can protect mitochondria from cisplatin in ovaries, we might do better. Obviously, there are many other things which one has to think about it.
Yeah, brilliant. Thank you. And we've got a couple of questions coming through now. So I'm just going to read them out. And again, either of you are very welcome to take it. So the first one's from Girish, and he's asking the efficacy of therapeutic plasma exchange, specifically plasma dilution during perimenopause, for diluting specific progerotic or pro-aging factors that drive ovarian aging during this critical time period. Is there any specific progerotic factors from the body that have been identified yet? I mean, I have absolutely no idea if.
No, I mean, this is the research we're doing, and honestly, yeah, if we could answer that question, we wouldn't be having this conversation. But yeah, this is. I mean, again, I'm biased, but yeah, this is, I think, where the field should be heading.
Got another one here. So any relation between IVF treatments and either early menopause or delaying it? So if you get IVF, will that cause an early menopause or delay it?
That data is just becoming available. So, IVF is only 40 some odd years old. So we don't know what the long-term effects of these hyperstimulatory cycles are. We don't know what the long-term effects of doing this to the mother's body are. And I mean, presumably there are going to be detrimental effects. We just don't know what they are or how big they're going to be.
Yeah. And actually, I have another question. Sorry to butt in for everybody on the chat. But this was something I kind of found in my research, and Jennifer, we've talked about it a little bit through email. But is there any relation to kind of the time that somebody has a child and their lifespan? So if I was a mother, my first child over the age of 35, is there a correlation with lifespan, or is that still a very complicated field that's yet to be understood?
Well, there's two things to unpack there, right? So one is that there's a very clear correlation between a woman's reproductive span, the age at which she goes through menopause, and her overall lifespan. Right, and it's a positive correlation. So the later a woman goes through menopause, the more likely she is to live longer, and the converse is also true. So that's clearly established. The question of the age at which you have a biological child, and then the number of children, that's complicated. But if you have a biological child later in life, that is basically an indication that your reproductive span is longer. And so in that case, that same correlation holds. But the question of number of children and when you have them, there's data out there. It's not entirely, I don't think it's crystal clear what direction it goes. And I don't know if you have anything to add to that, Dr. Singh.
No,
nothing for that one. That's okay. I mean, yeah, that was because I was reading quite a lot about that, and it seems very complicated.
It is very complicated, right? Because having a child is not a zero-sum game, right? It requires quite a lot of resources from the mother. So, yeah, and it also requires not just functioning ovaries, but also a functioning uterus. There's a lot of things that go into having a child that kind of extend beyond ovaries.
Yeah, definitely. And this is quite an interesting question. And I guess it's one that most women would maybe have. So what impact does hormonal contraceptives have on menopause and ovarian health? And I guess, Dr. Singh, you might know if that's got any impact on mitochondrial health and function in the ovaries as well.
So what I can say that the endocrine function is controlled by mitochondria. So all 4 steroid hormones go through some way or the other through the mitochondrial pathway. So I have a list of it here. Actually, is progesterone, mineralocorticoids, glucocorticoids, androgen, and estrogens. So any interference with contraceptives, with those hormones, going to likely affect mitochondria and vice versa. And this is pretty amazing. When I was doing some research before coming on here, I was amazed at how deeply the mitochondrial function is involved. On top of that, you find estrogen receptor localizes to mitochondria. So what that does to the mitochondria in relation to other functions, anybody guess? We showed last year the androgen receptor localizes to mitochondria. And we showed in 9 different ways, and people had hard time believing it. Even now, there are no believers, and but that's okay. I think you do more work and to prove that, well, So certainly, I don't see any. At least I'm not aware of the direct literature. Maybe Jennifer can allude more on it, but I'm not aware of the direct literature, which I can say. It's very likely.
Yeah, this is another place where we don't know. We're doing the experiments in real time in humans rather than figuring it out ahead of time in the lab. But yeah, we're doing this experiment now. We don't know what the long-term effects of contraception are on ovarian aging. There's probably clearly going to be an effect. As Dr. Singh said, steroid hormones are part of the biosynthetic pathways linked to mitochondria. And of course, I would also say, maybe just as a public service announcement, that a lot of women think that if they're taking contraceptives, especially oral contraceptives that keep them from ovulating, that they're somehow preserving the number of eggs they have, right? Because when you go through a menstrual cycle, you're losing about 1,000 eggs per month. And so I think many women erroneously think that if they're suppressing ovulation through oral contraception or patches or whatever, that they're not losing those 1,000 eggs. But actually, they're just losing 999 instead of 1,000.
That's right.
And so it's it's not in any way, shape, or form going to put off ovarian aging or preserve the number of eggs, which is maybe one strategy for delaying ovarian aging. Certainly, that's something that's being explored.
Yeah, definitely. Sorry, I've got quite a few questions coming in now, but we are coming to the end of the webinar, so let me just have a quick look. So there's one here that says some women have expressed that they feel their quality of life is better after menopause, kind of due to the unpleasant effects that are associated with the menstrual cycle. Would it be possible to preserve ovarian health in a state while mitigating the effects of the menstrual cycle, or preserving health of ovaries and inducing menstrual cycle when ready to have a child? Yeah, I think start simple. But on to you guys. What would you think about that one?
I think the answer is possibly, right? We so don't know. We're so lacking knowledge in this area that we don't know. But whether separating menstruation from ovarian aging is possible, I think probably it is. And to be honest, when women say things like that to me, I have to say, that the risks, it's a risk-benefit analysis essentially, right? And so you have to weigh the risks of going through menopause, the negative health consequences of being in menopause versus the benefits that you would gain from not being in menopause. And menstruation is something that no woman really enjoys. So if they are separable, that would be amazing, but we just don't know at this point.
Yeah, brilliant. Okay, so I'm just going to finish on one question. And it's kind of a nice compliment to you guys as well here, so it's a good one to end on. But what GCRLE, UVA, and other bodies are doing in the space is amazing, but there's still a lack of education out there. How can science better inform women what's going on in their bodies and why it's so important for their health and well-being, and equally important, ensure it doesn't just become a woman's problem? Great question. Either of you?
That's a great question. And this is what we're trying to do. I mean, we've published a white paper. I do these webinars all the time to try to spread the word about what we're doing. You now watching this are going to be our ambassadors. So it's your job now to go and talk about this as it should be a topic of conversation, I think, at every dinner table. It really does affect every single human on the planet. And directly, it will affect half the people on the planet who reach middle age. And indirectly, it's going to affect everyone. So it's something where we definitely need more outreach and more ambassadors. And we're putting out as much information as we possibly can, but we're also relying on you.
Definitely. So guys, thank you so much for joining me here today to talk about ovarian aging. It's honestly been a pleasure to talk to you both and learn about what you're doing. Just to say to our audience that if you do want to learn more about ovarian aging, we have the free report on ovarian aging on the Longevity.Technology website. And obviously you're very welcome to send any questions you have on to us after the webinar is finished. But thank you very much for joining us today. And we have another webinar on the opportunity of ovarian longevity as an investment from like an investment standpoint on the 26th of October. So if you would like to join us for that, that would be fantastic as well. But yeah, but for now, that's everything. And thank you so much, guys.
Thank you, Dara. And I'm happy to answer questions offline too, if anyone wants to reach out. Yeah.
Thanks, guys.
Okay.
Oh, thank you.
Bye.
See you soon. Bye.
Bye.